My name's Steve Meikle. I'm from the University of Sydney, and I'd like to thank the organisers for inviting me to give this talk about total body PET and the new collaborative opportunities that arise from it. So in conventional PET, we surround the patient with rings of detectors. Typically, they have an axial field of view of 20 to 25 centimetres. And if we want to image the entire body, we step the patient through the gantry, collecting data as the patient passes through the gantry. But this is a very inefficient way to collect all the available signal. In fact, we collect less than 1% of the emitted radiation. So in total body PET, the idea is to simply surround the entire body with radiation detectors, and that gives us up to a 40-fold gain in sensitivity. And it also gives us the ability to capture the kinetics of the radiopharmaceutical in every organ and tissue of the body. So that brings with it some new capability, both in terms of clinical and scientific applications. So clinically, it gives us better signal to noise ratio, faster scans if we choose to use the high sensitivity to do higher patient throughput, delayed imaging, so we can image for longer periods of time because of the higher sensitivity as the radiopharmaceutical decays, and lower dose. And the lower dose applications mean that we can apply PET in areas that we're not currently using it for in certain pediatric conditions, in maternal fetal medicine and studies of normal biology. Scientifically, it gives us the ability to capture pharmacokinetics from organs simultaneously, as I mentioned, and that's really useful if we're interested in studying systemic diseases. It gives us the ability to measure the arterial input function, no matter which organ we're imaging, in all studies, and so that's important for quantitative studies. It gives us the ability to measure signaling between organs, which could be important in, for example, neuromuscular conditions such as motor neuron disease, or in Parkinson's disease or diabetes. And it gives us the ability to track imaging targets over long periods of time, such as labelled cells and nanoparticles. And finally, it gives us the ability to accelerate clinical trials due to the ability to reach surrogate endpoints earlier because of the high sensitivity. So in 2015, a group of academics received a $15.5 million US grant from the NIH to develop this technology. And they partnered with industry to leverage those funds, which enabled them to build 3 systems on the West Coast and a 4th system on the East Coast. So on the West Coast, they built 2 primate total body imaging systems and a human total body PET that they called U-Explorer. And on the East Coast, the University of Pennsylvania partnered with Philips to develop the PenPET Explorer. So here are the first imaging studies from the East Coast system, the U-Explorer, collected in late 2018. And this is a male, 65 kilo male, injected with a fairly typical dose of FDG, scanned for 20 minutes, 90 minutes after injection. And you can see the quality of the images is very high compared with current state-of-the-art PET systems. And these data were published in the Journal of Nuclear Medicine in 2019. This is the first dynamic study to be performed on this system, showing the injection of FDG into a vein in the foot. And this is a time lapse over the first 30 minutes of a 1-hour dynamic scan, showing the tracer gradually accumulating in tissues that have high metabolic rates, such as the brain and the heart, and being excreted, as we know, through the urinary system into the bladder. And then what these investigators showed was that with this exquisitely high sensitivity, they could actually capture the really rapid dynamics in the first phase of the delivery of the tracer by framing the dynamic with 100 millisecond frames. And here you can see the tracer going up through the circulatory system, through the right side of the heart. You can see the heart beating, out through the left side, and then out into the periphery. So demonstrating the ability not only to capture the kinetics and use and do that with very high sensitivity, but also very high temporal resolution. So these systems have been commercialized, or at least one of them has. So the U-Explorer system is now offered as a commercial clinical product by United Imaging Healthcare. It has just under a 2 meter axial field of view, 3 millimeter spatial resolution, and time of flight resolution, which is fairly moderate at 430 picoseconds. And last year, Siemens announced at the European Association of Nuclear Medicine Congress, the virtual conference, a product of their own called the Siemens Biograph Vision Quadra, which is essentially 4 vision systems combined to give an axial field of view of just over 1 meter. And in all other respects, the technology and the performance of this system is similar to the vision, with 3.4 millimeter spatial resolution and excellent time of flight resolution of 230 picoseconds. So the detectors in this system are exactly the same as those in the vision. And these are some images off that system with the 106 centimeter field of view, showing the excellent image quality obtained with this system, even with a scan acquisition of 15 seconds, the images are acceptable. These are MIPS, by the way, not coronal images. But certainly at 1 and 4 minutes, the images are of a very good diagnostic quality, and at 10 minutes, they look extremely good. So around about the same time as the UC Davis and United collaboration were developing their systems, the Australian National Imaging Facility made a proposal to the Commonwealth Government to bring this technology to Australia, and that led to funding in the 2018 budget, an allocation towards NIF, which included $7.5 million towards a total body PET system. And this funding went to the University of Sydney, and we are partnering with Royal North Shore Hospital to deliver this technology and the National Imaging Facility. And that brings a whole range of collaborative opportunities, leveraging not only the total body PET capabilities, but also our capabilities in PET physics and radiochemistry to apply all of the that total body PET has to offer in terms of metabolism, signaling, and treatment response, therapy response, to address the many challenges in complex multi-organ diseases, such as the ones shown across the top here, and undoubtedly many others that are not shown here. So the current status is that the total body PET system will be installed at and operated by Royal North Shore Hospital in Sydney. The research access will be managed by Sydney Imaging on behalf of the University of Sydney and NIF. It'll be used 50% of the time for clinical studies and 50% for research studies. And it'll be operational in the second quarter of 2022, so about a year from now. Finally, I'd like to just acknowledge the people who contributed images and slides for this talk, the people from UC Davis, United Imaging, and Siemens. And I'd also like to acknowledge the funding from INCRIS via the National Imaging Facility to make this project possible, and also a grant by the University of Sydney and UC Davis to develop collaborations in this area.